because it is a general growth medium. So again, what that means is it has all of the nutrients it needs to grow, nothing to select or inhibit. So that's going to be our nutrient auger. So let's start by looking at mannitol salt auger, which is going to be the pink color plate. So in mannitol salt auger, we had a couple of different things we were looking for. First of all, this media has something called mannitol in it. I mentioned what mannitol was last time. What biomolecule is that? Okay, so it's going to be a sugar. So a sugar is going to be the mannitol. Okay. Now this sugar is going to make the media differential. What did it mean to be differential?
Complex.
Visual difference. Excellent. So remember, differential just means a visual difference. So what is happening here is some of the organisms that we're plating can eat mannitol. They like that sugar. They have the enzyme to eat mannitol. But other bacteria, they don't have an enzyme to eat mannitol, so they're not going to. Okay, so they're not going to be able to eat the sugar. So this sugar is going to make our media differential. And the reason it does that is because there is a pH indicator in here that's called neutral red. And neutral red is going to turn yellow if an acid is present. Turn yellow if acid is present. Okay, well how do I get an acid? Well, let's think about what we learned in glycolysis. When we start our dance in glycolysis and we go through glycolysis, what is the end result at the end of breaking down that glucose? What's my 2, 3 carbon molecules at the end?
Pyruvic.
Pyruvic acid. So I'm making an acid. So when I eat sugars or break sugars apart, I make an acid. Everybody with me on that? So if I'm able to eat the mannitol, I'm going to create an acid, which is going to cause my pH indicator to change colors. In this case, I'm going to turn yellow if acid is present. Does anybody see on their plate an organism who has turned the media yellow? Which one? Staphylococcus aureus. Good. So Staph aureus is able to eat the mannitol. How about Staph epi? Is Staph epi yellow? No. So Staph epidermidis is not able to eat the mannitol. So this allows me to tell a difference between the two species. If I have a pathogen of staph, it will turn yellow. If I have a non-pathogenic strain, it will stay white, which means my background stays pink. Everybody with me on that? Okay, so that's what's making it differential. But we also have another component. So I'll put the one here for the differential. We also have a component that makes it selective. So I mentioned last time there was something that made our media selective. I told you it was a really high amount of something, like 60 grams. Do you remember what it was? And it's in the name.
Salt.
Salt. Excellent. So what makes this media selective is a very, very, very high salt. Now, think about Staphylococcus. Staphylococcus grows on our skin, right? Is our skin salty? Yeah. When we sweat and when we're like working out and just everyday life, we have salty skin. We're a little salty every day. We're working on the saltiness. We're a little salty every day. So because of that, the staph have adapted to be able to live in high salt environments. So they are actually able to turn pumps on their cells on and off that can move salt in and out. Now, we tested 2 other bacteria on this media. We tested E. coli and we tested Proteus. How did they do on mannitol salts? Did they grow? No, we shouldn't see any growth. The reason we don't see any growth is E. coli and Proteus, they cannot handle the salt. So in this case, I'm selecting out organisms that can handle salt. And once I've selected separated them out, I'm making them look different. So I can tell pathogen from harmless strains. Does that make sense what's happening on this plate? If you're not sure, this is a good time to ask, but this is what we're looking for. Alright? Okay, so that gets us through our MSA. So the next plate we're going to look at is the EMB plate. And I told you this was eosin and methylene blue. So this one also has sugar in it. And in this case, it's going to have lactose and sucrose. Now, what we're going to be looking for with these sugar eaters then is who's able to eat both of these sugars and who is able to eat a lot of these sugars. In other words, who makes a lot of enzymes. This media is looking for a special type of organism group called the coliforms. And we're going to learn a lot about coliforms in the future. Coliforms are just bacteria that live in the gut of mammals. So would E. coli be a coliform?
It's just
Yeah, because he lives in our gut, right? So an organism that lives in our guts. How about Staphylococcus? Is Staphylococcus a coliform? No, because he doesn't live in our guts. All right, so if it lives inside of us, in our guts, in our intestines, then it's a coliform. That's just a group, okay? So in this case, we have sugar, we have lactose, and we have sucrose. Well, it turns out E. coli absolutely loves lactose. Like it's like me with Cheetos. It's like its favorite thing in the whole wide world. So it's going to eat a lot of lactose, okay? Now, if I'm eating a lot of sugar, what am I producing as a side effect, an acid or a base? An acid. So if I eat a lot of lactose, I'm going to produce a lot of acid. So I get a pH shift in here. So I also, in this media, have a pH shift. And in this case, we're going to be using our eosin as one of our pH shifters, which is in the name, eosin methylene blue. That eosin is going to detect when there's a shift, and it's actually going to create kind of a metallic look. It creates a metallic colony appearance. So when you look at your plates, does somebody have kind of a pretty metallic look? Yeah, which one is metallic looking?
E. coli.
E. coli, good. So that E. coli, who loves lactose, produces a lot of acid. And because of that, we get this really cool metallic shift. Okay, the other organisms that we set up on this plate, we set up our Staphylococcus, epidermidis, and Staphylococcus aureus. Did either of those grow? No, good, they shouldn't grow. And the reason for that is, remember, we have methylene blue in here. So methylene blue and eosin are also dyes. And I mentioned last time that dyes can affect the growth of gram positives. If I'm trying to build a thick lasagna layer and I am a gram positive, those dyes kind of get in the way and they make my layers slippery. And so my layers have a harder time stacking, and so it kind of falls apart. So if I put a gram positive on EMB, it is inhibited because of the dyes. So my sugar is going to be making it what? Is it a visual difference, or is it killing things? Is my sugar killing things?
No.
No, it's a visual difference, right? So does that mean it's selective or differential?
Differential.
If I'm not killing something, if I'm getting a visual difference, that's differential. So the sugar is going to be differential. You can see why we're going over this. It gets really confusing. So you want to like sit there and kind of look at this. So my sugar is going to be what is differential. That's going to give me a visual difference between the two. Who loves lactose? Who makes the most acid? Who causes a pH shift? All right. What's going to make it selective is going to be the presence of the dyes. The dyes, the eosin and the methylene blue, which is going to kill off any gram positives. Okay? So it's going to kill all gram positives. So with this in mind then, if I have any coliform on this they will grow. If I see metallic, that means E. coli. If they're growing and they're not metallic, that means a different background coliform. Okay? Yes?
What if it has like a greenish metallic?
Does your E. coli have a metallic color? It should.
It's Staphylococcus.
It shouldn't. So what did I tell you about the bacteria?
They have mission?
They'll tattle on you. Yeah, so if you're seeing growth and you're seeing a metallic sheen, that tells me there was E. coli on the loop. Yeah, because the staph, they cannot grow. They fall apart. So I shouldn't see any growth at all. At the most, occasionally, if I had a really thick amount of staph, I might see a little film where they tried to grow and then they die off. But I'm not going to see any color changes or reactions.
For E. coli.
Yeah.
For the manifold cell hugger,
Yes.
does that mean that the eosin kills the gram-positive bacteria in that case too? Or is that not?
Okay, so eosin's not in mannitol salt.
Or, I'm sorry, I meant neutral red.
Okay, so the neutral red is our pH indicator. That one does not kill things, it just detects the
This
pH.
is the detector, okay.
Yeah, so this one's a little different. That dye that we're using is also a pH indicator. So he's doing a double
Oh, I
duty.
see, okay.
Yeah, so he's a pH shift and he's a dye.
Okay.
Yeah, good question. Okay, are we doing okay so far? I know it gets confusing, that's why we're going through these, we're spending some time. Okay, the next one is McConkey, and that's gonna be kind of a violet color. I don't know, like, what color can I describe it? Yeah, it's kind of, it's this, like, I don't know what you're about, it's this violet kind of pinkish color. Okay, so McConkey is always set up with urine cultures. If you work in a lab and somebody has signs of a UTI, what's a UTI?
Urinary tract
Excellent,
infection.
urinary tract infection. If somebody has a UTI, we are always gonna set up a urine sample on McConkey. It's one of our cornerstones. Kinda like how we always do a gram stain, we're always gonna throw this on McConkey. Put a drop of urine, spread it out. The reason for that is probably about 95%, and again, you don't have to remember this, but about 95% of UTIs are E. coli caused. That's a pretty high number. So majority of our UTIs are E. coli. We do have a few oddballs, but for the majority. So McConkey's gonna be another one that's gonna help us find E. coli. McConkey's gonna be similar to EMD. The only difference is instead of having lactose and sucrose, it is going to have just lactose. So it is going to be differential because of the sugar lactose. Okay, so it's going to just have lactose inside of it. Now, it's also going to have a pH shift. It's going to be using neutral red as its pH shift. So it's going to be using, it's neutral red. You know what, and I just realized, I gotta fix this. This should be phenol red. Sorry, guys. That should be phenol red. All of a sudden, I'm like, wait a minute. If I'm neutral red over there, that's gotta be phenol red there. Okay, so pH indicator here, phenol red. pH indicator here, neutral red. Okay. So in this case, I'm getting a difference based on if I'm able to eat the sugar lactose. But this media is a little different. If I can eat a lot of lactose, I make a lot of acid, so my color is going to shift to a pink colony. So look at your E. coli. Does it have a lot of pink colonies? They almost look like Pepto-Bismol. Yeah, so like that's like the distinctive E. coli color, is they have this pretty pink colony. How about Proteus? What color does Proteus have? Is it kind of yellowish? Like a little bit clear? Because I kind of Proteus. Yeah, kind of like a clear color, right? Didn't really do a whole lot. Do you have your Proteus?
Here's a pink.
I want to see what Proteus looks like. Right here. Yeah, so this is Proteus.
That's pretty good.
Do you see how he's kind of yellowish? Yeah, so not really very exciting, kind of actually changed the media. So what that means is that my Proteus cannot eat a lot of lactose. So if I see him there, it's not causing a pH shift, okay, because he's not eating a lot of lactose. It can grow, but I don't see a color shift. But no color shift, okay? E. coli can grow, and it is going to have that pink color shift. Now, how did our Staphylococcus do on this plate? Did either of the staphs grow? No. And hopefully that makes sense, because remember, MacConkey has some dyes in it, right? What was the dye that we mentioned was in it? We've used it before.
Neutral red.
So not neutral red, that's a pH indicator. Remember, it has crystal violet. It has some crystal violet in it. So crystal violet is going to be a dye that can be in the media, and again, this dye is going to inhibit gram-positives. So if I'm a gram-positive, I'm not going to be able to grow on this plate at all, okay? So no gram-positive organisms. Okay, just to make sure we understand what's going on. What is making the media differential? Making it look different? The sugar, and specifically lactose. That makes it look different. What is making it selective? The dyes. Excellent. So it makes it selective because of the dye. So when you're studying these, the nice thing about these is MacConkey and EMB are very similar. They're very similar. The only difference between the two really is the type of dyes and the fact that this one has lactose and sucrose and this one just has more lactose. All right, so that's really the differences that we're looking for. All right, so hopefully this makes sense as we're going through. Now, questions that I like to ask. What makes MacConkey selective? What makes MacConkey differential? What makes mannitol salts selective? What makes mannitol salts differential? What should Staphylococcus aureus look like on mannitol salts? What should Staphylococcus epidermidis look like on MacConkey? It shouldn't grow, right? Those are the kinds of questions you want to ask yourself as you're studying. Making sure that you're understanding why we're using the media, what it's doing for us, and how it's helping us, all right? So based on these results, which of my organisms are gram-positives?
MSC.
The staph.
Staphylococcus.
Yep, so Staphylococcus are going to be my gram-positive. Who are my gram-negative? Most of species here. Who are my gram-negatives? E. coli's one, excellent. Who's the other one? Proteus. Proteus is the other one. So Staphylococcus aureus and Staphylococcus epidermidis are both gram-positives. E. coli and Proteus are both gram-negatives. How do I know this? I haven't done a gram stain. What do we talk about here? The dyes. The dyes are going to kill all the gram-positives. So if I see growth, that tells me it's a gram-negative. If it's not growing, that tells me it's a gram-positive. See how we can use this? It kind of helps us, right? So this is a way that we can verify that our original gram stains were correct. If I did a gram stain wrong, and let's say I thought a gram-positive was really a gram-negative, maybe I over-decolorized, and I put them on an EMB and a MacConkey, thinking that it's a gram-negative, and I come in the next day and it doesn't grow. What does that tell me about my gram stain? That I was wrong. And that tells me I need to go back and do it again. Do you see how that works? So these media confirm our results. Remember, I told you with the gram-positive and the gram-negative, it tells us which path to go down. It's like you come to a fork in the road, and if I'm gram-positive, I go this way. If I'm gram-negative, I go this way. If I choose the fork incorrectly, I'll figure it out pretty fast because I'm going to see some funky results on the media going forward. All right? Okay, so hopefully this makes sense. Do be sure to take pictures of the board before you leave today. And you'll be able to look through the results again. It's all outlined for you on page 60 and page 61. It's also explained in the intro on 59. So this is like the crude notes, but you'll want to look at those two pages. For your data, on page 62 and 63, you're going to record your results. So let's look at the first one together, mannitol salt. The first column says growth, and this is yes or no. So looking at mannitol salts, did Staph aureus grow?
No.
Yes, so we put a Y there. Did E. coli grow?
No.
No, so we put an N there. Staph epi?
Yes.
Yes. Proteus vulgaris?
No.
No. Then we go over, if growth is present, record the color of the colonies. If absent, write NA. So for E. coli and Proteus, we put NA. For Staph aureus, is growth present? We said yes. So now we're gonna write what color was the colony. So what color was Staph aureus?
No.
Yellow, excellent. So we'll write yellow in there. What color was Staph epi? He was white/pink, right? Because he didn't change the media, so white, pink. Then it says record your gram reactions. Well, we just did that on the board. We know if it didn't grow, it has to be a gram negative. Yeah, so we'll put gram negative for E. coli and Proteus. And for the Staph aureus and Staph epi, they are gram positive. So that's what we're going to be doing for all of your media. All right? Okay, so those are our results. And then, like I said, I know it's a lot. It's something you have to kind of sit there and look through. All right? And kind of like make sure that you're understanding it. All right, well, let's talk about what we're going to be setting up today. So we're going to be setting up Lab 10. Lab 10 is going to be looking at extracellular enzymes. An extracellular enzyme, does that sound like it's in the cell or outside of the cell? Outside of the cell, good. So the idea behind extracellular enzymes is sometimes as a bacterial cell, I have lots of food around me, but it's too big to bring in through my cell wall and my plasma membrane. It's kind of like if you go out to eat at a steakhouse, like if you're a millionaire nowadays, because I swear restaurants are so expensive, right? I had sticker shock the other day at McDonald's. I took my kiddo and her bestie to McDonald's. It was $32. I didn't even get food. It was like for two teenagers, it was $32. I don't have McDonald's money anymore. I don't know. Like, this is the end of McDonald's. We're done for a while. We'll try Taco Bell. Maybe that one's better. I don't know. I haven't tried it. So yeah, if we could buy a steak and I put that steak down in front of you, okay, are you going to be able to eat that steak as is, or do you need to use like a fork and a knife? Hopefully you use a fork and a knife. I don't want to go to a restaurant and see somebody pick up a steak and be like, like caveman style. We don't want to see that, right? So that's kind of what's happening with extracellular enzymes. I'm a cell, and around me are steaks, but I can't bring them in because they're too big. So I release enzymes into the environment. My enzymes are like a fork and a knife. They're going to cut the steak into bite-sized pieces. Now that they're bite-sized, I can pull them into the cell, past the cell wall, past the plasma membrane. Okay, so that's what I'm looking for. So we're going to be setting up 3 different types of media today to look for extracellular enzymes to see if our organisms have them. Now we are skipping exercise 10B. We were not able to get any lipase media. But what kind of molecule do you think lipase would be breaking down? Lipids, yeah. So I do still want you to know that lipase would break down lipids. We're not going to be able to see it, and that's okay. They were back-ordered on the media. But just know lipase means it can break down lipids. All right, so to do our experiments today, we're going to be setting up some starch agar, some urea agar, and some gelatin agar. Now, be sure to read the organisms that you're setting up with each of them. So for the first test that you have is 10A, it's the starch agar plate. You're going to be setting up Bacillus and E. coli. Everybody's going to be setting up a starch plate. So I'm going to take a plate and I'm going to do exactly what I did last time. I'm going to put a line. I'll have E. coli on one side. I'll have Bacillus subtilis on the other side. And I'm going to do a fishtail. So remember how we do that. We do a straight line down and then a squiggle. And a straight line down and then a squiggle. Making sure we don't touch the edges or the other bacteria. Okay, so that's what we're going to be doing for our starch plate. We're going to be looking to see next time who has the enzyme that's able to break down starch. So starch is going to be a very big biomolecule. Is it a sugar? Is it a protein? Is it a lipid? What is it? Carb. Good. So it's going to be a carbohydrate. So one of these bacteria can produce an enzyme called amylase. Now I know we said that usually enzymes make sense, so if we think about this, it should be starchase, but starch actually used to be called amylose. That was its old name. And so its sugar, its enzyme that would break it down was amylase. We later on changed the name to starch, but we didn't change the enzyme. So it's like a throwback, all right? So we're gonna be looking to see who can create amylase. So we're gonna grow our plates, and then next time we're gonna do a test, and if our starch is broken down, we're going to see a different reaction than what we see if they could use the starch. And we'll go through that together next time. Because do you guys like going through the things together when they're in front of you? I can do it however you want. I could just give you the plates back and have you interpret them, or if you prefer, we'll go through them together. Which do you want to prefer? Together. Okay, all right, so we'll keep doing it that way. I know it makes the intros a little longer, but I feel like it kind of helps, right, to kind of understand. Okay, you're also going to be setting up a urea slant. So remember, the slants are the ones that have kind of a slope to them. And the way you're going to set that up is you're going to put your loop to the base of the slant. We're not sticking into the butt, we're just going to the base of the slant. We're gonna drag upwards, and then we're gonna go back and we're gonna zigzag. So we get a nice amount of bacteria on our slants. With these slants, we're gonna be setting up our E. coli and I believe our Proteus. There's my slant on here. Yep, our E. coli and our Proteus. And then the last one that we're gonna set up is our gelatin deeps. So these are going to have a very deep tube associated with them. And by the way, for the urea, bless you, for the urea agar, you're going to separate that. One of you is going to set up E. coli, and then the other lab partner is going to set up Proteus. Okay? I think it's Volcaris, right? Is that the one I have you guys set?
Yeah.
Okay, cool. All right, so one of you does E. coli, one of you does Proteus. Alright, so you guys can figure out how you want to do that. The gelatin agar, the same idea. One of you is going to set up Staph aureus, the other one is going to set up Staph epidermidis. Now, the way you're going to set up your gelatin is we're going to use a new tool. Up until this point, everything that we've used has been a loop. In your drawers, though, you have a needle. So it looks just like a loop, it just doesn't have a loop on the end. So it looks basically like this, and then it's just got a straight point to it. That's your needle. And the way you're going to use this to set up is you flame sterilize your needle, just like you normally do with a loop. When it's cooled, you take your plate and you take the needle and you roll it in the bacteria. So you pick up a lot of bacteria on the tip. So all I'm doing is I'm holding that needle and I just roll it on top of the surface of the plates or the tubes, whatever you guys have in front of you to get plates. And I'll pick up a little bit of gunk on the surface of that needle, okay? And then what I'm gonna do, this is so satisfying, is you get to stab the heck out of the gelatin, okay? So you get your needle and you're gonna go straight in and straight out. Not all the way to the bottom, but about 3/4 of the way down, okay? So I have a little gap at the bottom. So I stab and I lift. And then I stab and I lift, and then I stab and I lift. And I'm gonna keep doing stabs and lifts in different areas. I like to picture the psycho music when I do this. Oh, it's so satisfying, right? Or whoever's really bugging you at work, you picture their face in the meeting. You're just like, ah, yeah. Really, you just get all of that aggression out. You're gonna do a stab 6 times. You have to stop at 6. I know we wanna keep going because we have a lot of aggression built up, but we wanna stop at 6, all right? And I'm not stabbing in the same spot. I'm moving around in the media. What I'm doing is I'm putting a lot of bacteria in there. Now, I don't have to keep going back to the plate. When I pick up all of those cells to where I can see the paste, I have plenty. So each time I'm stabbing and lifting out, I'm dropping off a lot of cells. Don't worry, there's a lot there. Now, another thing I want to keep in mind is if I'm stabbing to 3/4 of the media, I need to make sure I flame sterilize my needle long enough. Right? Not just the tip, but long enough to cover how deep it's going to go into the media. Okay, once I'm done with this, everything's going to get labeled. Remember, our tubes get tape on them for the labeling, and they're all going to go into the incubator. Now, with my tubes, I want both of them to have loose caps. So remember, that means that they're not falling off, but if I can pick them up and I can shake them, I can hear the jingle. Okay, I should hear a jingle, but they're not gonna fall off. Why do I want loose caps? What's getting inside?
Oxygen.
Oxygen. So there's only a very rare types of media that we tight cap, and I'll always tell you when we tight cap. For all of the media that we're going to be working with in these next few labs, we're going to loose capping. All right? We're going to put all these guys in the